Hello, my name is Lucian Williams, and for my Exam 3-1 video, I will be describing the replication fork. The spot on the DNA where replication begins is called the origin of replication. Prokaryotes only have one of these, while eukaryotes have multiple ones, each producing two replication forks that proceed to either side. In order for replication to occur, the two parent strands of DNA have to be separated, which is done by helicase, which breaks the hydrogen bonds between the nucleotide pairs. These newly separated strands are then bound to by a single-stranded binding protein, which prevents degradation while they're separated and prevents the two strands from rebonding together. DNA gyrase binds above the replication fork, and this helps the replication process by relieving torsional strain that occurs when you unwind a double helix, preventing the rest of the DNA from supercoiling. The formation of new strands of DNA is begun through RNA primase, seen in red. RNA primase creates RNA primers, seen in blue. These RNA primers are then added onto with DNA nucleotides, which extends the DNA molecule. The addition of DNA nucleotides is done by DNA polymerase III. DNA polymerase III can only act in the 5' to 3' direction. Because of the directionally restrictive nature that DNA polymerase III adds nucleotide bases, you'll see two distinct environments on the two strands of DNA that are being created. One such environment is the leading strand. This is the strand of DNA whose 3' base is proceeding in the direction of the replication fork. Because the 3' base end is continually being expanded, the DNA polymerase III can continuously add DNA bases uninterrupted. On the other strand, it's the 5' end of the new DNA that's being expanded, meaning that DNA polymerase III has to continuously stop and restart DNA replication, creating what are known as Okazaki fragments. Okazaki fragments are the short, discontinuous segments of DNA on the lagging strand that need to be bound together to create continuous DNA. At this point, we still have RNA primers at the beginning of the continuous and discontinuous strands. DNA polymerase I is responsible for removing the RNA primers and replacing them with DNA nucleotides. DNA polymerase I, however, fails to bind these new nucleotides to the adjacent strands. Ligase comes in and it fixes the phosphodiester nicks, creating continuous strands of DNA.